Okay, so is it visible? The slide is visible. Transcriptomics.
Yes, sir.
Okay.
Yes, sir.
So we will start from the very basic idea that how can we detect and quantify the RNA which are present inside the cell. So most of you know about the details of this technique because already you have studied that thing, right? So can you say some points on this? What are the different detection techniques?
Like blast.
No, this is not bioinformatics. I am asking for experimental techniques, experimental methods.
Sir, southern blot.
For
Yes, sir,
RNA,
southern.
for RNA, you will do southern blot. For RNA, I'm not saying DNA or I'm not saying protein.
Sir, northern blotting.
Northern blotting. And any other technique?
Sir, UV তে 260 to 280 nanometer measurement RNA quantity.
Okay, that is good. RNA quantification. And any other method? See, why I am asking this? Because this transcriptomics will start from this RNA sequencing, okay, or RNA detection. So that's why you should know that what are the limitation of the previously identified techniques, so that this kind of advanced techniques are required. Okay, so you know that omics technologies are very advanced uh, techniques. Nowadays, most of the labs are doing different types of omics techniques. But why this kind of omics techniques are required? So that is the problem. That is the question. Because earlier also uh, they could use different techniques for identification of RNA, but suddenly why they are interested to such kind of techniques, modern techniques? So for that, I would like to know that what are the previous techniques scientists usually used for detection of RNA. So one of them are northern blots, right? So what is done there? An RNA sample is digested into the fragments and they are separated by their size in some gel electrophoresis technique and then transferred to a membrane, right? And that can be probed to the test for the presence of particular or hypothesized RNA transcript. Whether that is present or absent, you can detect that thing using northern blot, right? And you can also measure the abundance of northern blot, abundance of the RNA using this northern blot technique by checking the thickness and color of the band in the gel electrophoresis. Okay, so it could be possible to detect RNA in this technique. Well, then what are the other methods available? You are saying UV detection method and quantification of that RNA in under 260 to 80 ratio. Okay, that is one thing. Any other technique? Those who have done in laboratory work in any project on this RNA, they have you used PCR? Anyone?
Yes, sir.
Okay.
Done. I have done PCR.
Okay, so can you use PCR to detect RNA? can you use PCR to detect RNA? These are very basic questions.
No, sir.
No,
Sir, RT-PCR is also quantifying RNA, viruses RNA.
right. Not only virus, you can check any kind of RNA using RT-PCR, right? Not only for virus. You can use plant RNA, you can use animal RNA. Where is the problem? So, what is the principle of this RT-PCR? Can you briefly explain? Anyone? What is going
Sir,
on?
yeah, from RNA to there is a reverse transcriptase, I think. So reverse transcriptase will transcribe DNA to RNA and then amplify. RNA to DNA, sorry, sir.
Okay. So, you are saying that what is reverse transcriptase?
The reverse transcriptase is an enzyme.
Enzyme. Okay. Which can transcribe mRNA to
mRNA to DNA.
DNA. Right. And why do we need this step?
Sir, because in polymerase chain reaction, the DNA can be amplified. DNA can be amplified. The growing chain will add the DNTPs in the DNA chain. So we need to if we need to amplify the RNA, we first have to change RNA into DNA.
Okay, so you are saying that the DNA can be amplified by normal PCR, and that's why we are making the RNA to DNA. But why not? We can use RNA as a template, and we can synthesize more RNA on that using the same technique. Where is the problem?
স্যার যে ট্যাগ পলিমারেজ থাকে ওটা আরএনএ কে অ্যাম্পলিফাই করে না, ওটা বেসিক্যালি ডিএনএ কে অ্যাম্পলিফাই করবে।
But why? Because AUG
The
all
RNA
are there.
RNA is a single stranded. That's why.
Yes, that is one point, and more importantly, due to this single strandedness, this RNA are very much fragile. Okay, DNA is more stable than the RNA, and working with RNA is very much difficult. Okay, so and the thing is, PCR requires two primers, which will synthesize the target DNA. Okay, in both the strands. So in case of RNA, there will be some problem for making such thing. Okay, so that is reverse transcription or reverse transcription PCR or RT-PCR using reverse transcriptase. Now the question is, so northern blot or RT-PCR or UV visible spectrophotometry you can use for detection and quantification of RNA. So next, why this kind of high throughput technologies like microarray or RNA-seq techniques are evolved? Okay, so you have heard about microarray technique or RNA-seq technique to quantify the RNAs in a cell. Now question is, why? What are the requirements for this kind of large scale analysis? What do you think?
Sir, microarray is specifically used to quantify the protein expression.
Hmm? No. There are different kinds of microarray. That microarray can be used for protein also, but most of the microarray from the very beginning, they are used to quantify the RNA. That is for transcriptomics analysis. Okay, but nowadays this kind of protein chip microarrays are also designing, and well, but the question is why this kind of arrays are required. We can do that using northern blot or reverse transcription PCRs also. So why microarray? Why RNA-seq? Because these are the costlier experiments.
So the time is a factor. I mean, the PCR, it requires more time.
No. These are only how much time the PCR needs. Who have done that? He can tell you. Who just now who told that you have done PCR? So what is the time requirement for one run? Can you tell me, or you forgot? Okay.
I forget, but I think for the amplification we need 1 minute and another is 45 seconds.
For each cycle. Okay.
Each cycle, yes sir.
Each cycle. So approx 1 minute or 1.5 minutes. So 30 cycles will required about 1 hour and 1.5 hour. Okay, it depends on the other parameters also, but you can finish it within 3 hours. So it's not a big deal with RT-PCR technique. Okay. So what could be the reason? Are you thinking, all of you? Any clue?
Sir, as the result, the DNA will amplify and
No. Why the DNA will amplify? I'm not getting you. Where the DNA will amplify? You are trying to say? Can you elaborate? Can you explain? What are you saying?
sir, I didn't understand your question, sir.
The question is very clear. Question is you can identify RNA using the other two methods like northern blot or RT-PCR. Now suddenly from the last decades you have seen that microarray techniques or RNA sequencing techniques are available in the market, and most of the scientists are using this kind of techniques. Now the question is why they have jumped from the old techniques to the newer advanced techniques.
Sir, accuracy and time consuming.
So which one is time consuming?
So northern blot is time consuming.
Okay, northern blot is time consuming.
And in the northern blot, we cannot quantify a large number of DNA where is the RNA, but in the microarray we can quantify a large number of RNA in the same time.
Right, that is the point. That is the point. So earlier the scientists were focused on a particular gene, okay, or a particular RNA. So that's why, or maximum two or few RNA at a time. Okay, and that's why these kind of techniques were good for them. Okay, but when they require more number of genes or more number of RNAs to be quantified, so it was not easy by using northern blot or RT-PCR kind of technique. Okay, so that is the main reason. We cannot analyze more than 96 samples at a time using the PCR sample. You need multiple PCR for doing that thing. Okay, but if you are using microarray or RNA-seq techniques, then you can analyze a number of samples, a number of genes, a number of RNAs at a time. Okay, and that's why these are more powerful. These are more powerful. Now, with this, you should remember that even if you are performing large scale analysis using microarray or RNA-seq, now what is your goal? Obviously, your goal is to identify some hits, some major proteins or some major DNA, major genes in your sample, so that you can say that okay, the control has not expressing that gene, or the treated samples or the disease condition sample are expressing those genes. Okay, so this kind of analysis you can perform, and from that you are trying to get some hits, some molecules by which you can further stop the progression of that disease or treat that patient, isn't it? Because from the 20,000 genes of human, you can find out 4 to 5 genes which are controlling some disease. Say for example, okay, which are very much prominent, which are very much influential rather than all other genes to cause a disease. Okay, now even if you are finding out this 5 to 6 genes from any sample, then also you need to experimentally verify that whether that gene is really causing that disease. Okay, because the one problem with this kind of large scale data are they are not so much accurate. Okay, their accuracy is good, but they are not so much accurate what you could do in RT-PCR or northern blot technique. Okay, so again you should recheck those gene expression by using RT-PCR or any other small volume technique. Okay, am I clear what I am trying to say? Okay, so if you understood this, then I'm going to the next topic. Now, I told you what about the omics technologies. Now, this microarray and RNA-seq, these are the examples of omics techniques, right? These omics techniques actually are required uh, to be high throughput. Now, these are different terminology. You should understand these terms first. So, what do we mean by high throughput? What do we mean by high throughput? Have you heard about this term earlier?
Yes, sir, we heard, but we don't know exactly what does it mean.
Okay. So actually high throughput means they need to analyze very large number of genes or gene expression or protein expression either in a single procedure or a combination of the procedures. Okay. So that means you think like this way. You are trying to analyze the whole sets of genes which are expressing at a particular cell. Okay, so what you are doing, you are isolating the RNAs, you are converting it to cDNAs, you are performing some microarray or RNA-seq, and you can identify that which genes are expressed at a particular condition. Now when you are collecting the sample, the sample volume is very less. that is using one appended tube, you are collecting the whole sets of RNA from a cell. But when you are analyzing the data, what you are retrieving from that particular sample, so the amount of data is very large. Okay, so that's why it is called high throughput techniques. In compared to the input, where the output is very large, so then you can think of that kind of technique are high throughput techniques. Okay,
Sir, please repeat this once again.
which one?
Sir, this high throughput.
Okay, I am saying that if you are trying to isolate or identify the genes which are present or which are expressed at a particular cell line. Okay, so what you will do, you will isolate the RNA from that sample, and then you will make that cDNA, and then you amplify that cDNA, and then you will do some RNA-seq techniques or microarray, whatever. Now ultimately what you are gaining, you are getting that whole sets of genes which are expressed at that particular cell line, isn't it? But think about when you are taking the sample, when you are collecting that sample, so that sample volume is very less. It is almost 2 ml, okay, in an appended tube. So using that, you are analyzing the data, you are analyzing the sample, you are identifying the RNA, and you are analyzing the sample by some computational tools and techniques. And when you are actually getting the long list of genes which are expressed at that particular cell line, that is huge. The number is huge. Suppose in that condition, out of 20,000 genes, suppose 12,000 genes or 16,000 genes are activated or expressed. Okay. So that means your input data, input volume, input sample is very less, but the output result is very high, large amount. And that's why these kind of techniques are known as high throughput. Okay, high throughput technique. Okay.
Okay, sir.
Now these omics technologies utilizes this high throughput screening techniques to generate the large amount of data required to enable a system level understanding of correlations and dependencies between the molecular components. Now again, there is a new terminology, that is systems level. Okay, because these omics technologies or high throughput biology, whatever you are saying, so these are again related to systems biology. Okay, I am not going to describe the systems biology now. I would like to focus on this omics technology. Now, this omics actually use as a suffix. Okay, suffix, because you will see that multiple omics technologies are available now. We will see in a minute that genomics, transcriptomics, proteomics, metabolomics, phenomics, multiple types of omics technologies are. Now, these phrase omics actually signify the measurement of the entire complement of a given level of biological molecules and information. Okay. So this omics technology actually providing the entire complement of a given level of biological molecules and related information. And that using that, a variety of new technologies will help us to explain the normal and abnormal cell pathways or networks of processes by simultaneous monitoring of thousands of molecular components. So just now I have explained that when you are focusing on some kind of experimental setup, suppose you are trying to analyze some pathways or some molecules which are controlling the cancer. Okay, cancer is a disease and which is not due to the activity of a single gene, you know very well, and different genes inside the cell actually form a network, and that network, gene network, actually controlling the cancer progression. Now, if you are trying to identify such kind of molecular network, such kind of gene network, so what you will do? You will take one control sample, you will take one cancer sample, and then you will try to identify which genes are expressed more or less in cancer sample in compared to control sample. Right? That is the normal way. Now, when you are focusing on multiple genes or multiple proteins in a sample, so then you need to take help from this kind of omics technology. Okay, so that is the reason. And to do that, even if you are doing biotechnology, so you are thinking that only the experimental techniques is sufficient for getting the information, you are absolutely wrong, because these kind of techniques are actually generating lots of data. Okay, and that data should be analyzed very precisely, very minutely, so that you can identify the meaningful information from that data. And that's why you need to take the help of bioinformatics also. Okay, or computational biology. And for that reason, biotechnology and bioinformatics are not separated nowadays. Even if you are doing the experimental techniques, you should know basics about bioinformatics, and even if you are doing bioinformatics, you should know about the basics of experimental techniques. Otherwise, both of you cannot communicate to each other. Okay, so the concept has been changed, and maximum lab nowadays recruits bioinformatician to analyze the data which are generated from different uh, large scale techniques. Okay. Now we will focus on different types of omics technologies. Okay. So the font is very less, but to make it in full screen, I need to uh, paste this full diagram actually. So you can see that. Is it visible?
Yes, sir.
Okay. So you can see that there are a number of technologies available, and which are actually analyzing the different layers of data or different levels of data. We know that we have genomic level data, or we can have epigenomic levels of data, or transcriptomics data, proteomics data, or metabolomics data, or phenomics data. For analyzing such different kinds of data, we have different technologies. Okay, so you can see that array based technologies, or DNA-seq, whole genome sequencing, whole exome sequencing, targeted sequencing, single cell DNA sequencing, Sanger sequencing, all these are related to analyze the genomic data. Similarly, there are multiple technologies available for epigenomics or transcriptomics or other data. Okay, now you know this technology will provide you the raw data set, and from that raw data set you can identify different things, like from the genomic data you can identify which are the SNP or which are the CND, or what are the indels. From a transcriptomics data you can identify what are the mRNA or what are the microRNA and their level of expression on that particular sample. Okay, and for doing that, for performing this kind of experiments or bioinformatics analysis, you need to consider something, and I'm not going to discuss the details of these things right now. you can search for different research and clinical applications based on these genomics data or transcriptomic data or proteomic data. So there are different references given in this paper, this article. But what I am trying to say that different omics technologies are available and they are very much useful to analyze different sets of data. Okay, and you will get different kinds of output from that particular data as per your aim or your experimental goal. You can choose which kind of data you will take, which kind of pipeline you will use. Now, what is pipeline? Pipeline means the steps to be followed to perform the bioinformatics analysis. Okay, so these are not one step process. You need to perform multiple steps for analyzing such large scale data. So based on that, you can get different outputs from different data sets. So that is my point. Okay. Now the question is, what is transcriptome? We'll focus on transcriptomics only. Okay. So what is transcriptome? I think any of you can tell me what is transcriptome. Very easy question. Anyone? What is transcriptome?
So transcriptome is as a whole the total RNA, I mean RNA of different proteins.
Total RNA.
Yes. Derived from different genes. So it's as a whole. I mean the study of different kinds of studies and analysis of RNAs, different RNAs.
So see, you are saying different things. Okay, different time. You are saying that this is the complete sets of RNAs. Again, you are saying that this is the complete set of RNA which are forming the protein. Is it so?
No, sir. No, sir. RNA not necessarily constitute proteins.
Right. So that means it is the complete set of transcripts. Better term is transcript. And based on that, it is transcriptomics. Okay. Transcripts means it can produce protein or it cannot be. Because different types of RNAs are available, like tRNAs or rRNAs or microRNAs or snRNAs. So those are not going to produce or synthesize proteins, right? So better to use the term transcripts in a cell and their quantity for a specific developmental stage or physiological condition. So that is the transcriptome. And the understanding of this transcriptome is very essential for interpreting the functional elements of the genome and revealing the molecular constituents of cells and tissues, and also for understanding the development or diseases. Okay. Next, what is transcriptomics? What is transcriptomics?
Sir, the study of transcriptome.
Okay. Study of transcriptome.
The transcriptome is a bunch of mRNA sequences. Study of mRNA is called the transcriptome mix.
Yes. So transcriptomics, which includes many types of RNA, and when you are studying the whole set of RNAs present in a sample. So that techniques will be termed as transcriptomics. So this is an omics techniques, transcriptomics, which includes the not only the messenger RNA, that is mRNA, it could be different types of RNA. Okay. Now, what are the scope of this transcriptomics analysis?
So transcriptomic analysis is very essential and important to connect between different species and to dig out the cues or the missing cues of evolution. Like we can analyze the tRNA and we can say that the echinoderms and the hemichordates are very alike, despite of despite of having a very different morphologic character. So these are the scopes of transcriptomes basically, and also transcriptomics is very essential, I guess, to diagnose some disease, genetic disease particularly.
Right,
Sir, also abiotic and biotic stresses can be found using transcriptomics analysis.
right. You can do different kinds of experimental setup using these technologies. So basically, you are trying to identify the specific subset of transcripts which are essential or which are important, which are playing important role in a particular cell type. Okay, so you are trying to identify the specific subset of a transcript. Say for example, in our genome we have 20,000 around genes, and in a particular disease condition you are trying to identify a subset of that gene, say for 10 genes or 5 genes. So you are trying to identify which are more influential to cause that particular disease. Okay. Now, there are certain differences with other techniques like genomics. Now, genomics depends on DNA, right? And which is approximately stable. You know, the DNA is not changing so fast, but when you are considering the RNA, the expression of RNA is a dynamic process. Okay, when you are studying the quantity of RNA across the time period, so you will find that RNA level may decrease or may increase across the time span. Okay, so you need to focus on that point also when you are trying to set one experiment based on transcriptomics analysis. So you need to focus on the time period also, because that is one major point. Okay, and this transcriptome can vary from different experimental environmental condition also, right? So that should be remembered. And the expression levels of the RNAs are also not fixed. Okay, that is also dynamic. Expression level of any kind of RNA inside a cell is also changing, right? So this kind of transcriptomics analysis are also known as expression profiling. Many times you will hear that expression profile of this sample is this. So expression profiles means the gene expression profiles, and that means the transcriptomics. Okay. Now, in short, if we are trying to identify the aim of the transcriptomics, so we could say that we can list out all the transcripts, including the mRNAs or non-coding RNAs or small RNAs, by performing such transcriptomic analysis. Okay, number one. Number two, we can determine the transcriptional structure of genes in terms of their start site or the end site, UTR site, or splicing patterns, or other post transcriptional modifications also. Okay, so this could be possible when we are focusing on a or a particular transcriptomic study. So from that we can find out some genes, and we can define the structures of the genes based on their uh, transcriptomic study. Okay, because we can find out the RNAs, and we can reverse the process. We can map the RNA to the genome, and from that technique we can identify that which genes are coming from which region of the DNA, and what is the structure of that particular gene which is transcribing that particular RNA. Okay, that kind of techniques are available, and we can do that. And we can also quantify, and we can also compare the expression levels of each of the transcripts present in a sample during any disease condition or any uh, stress condition. Okay, so that is known as differential gene expression. When we are talking about differential gene expression, that means we are trying to compare the expression levels of the genes in a condition with respect to a control. Okay, so some genes may be upregulated in one condition, or some genes may be downregulated in that sample, but for that you need a control. Okay, if you can set one control, then only you can say that with respect to that control, the genes which are overexpressed or which are underexpressed in a sample, in a condition. Is it clear? So that kind of thing you can perform using transcriptomic technique. Okay, got it?
Yes, sir.
Okay. So next, we'll focus on the different HTS platforms and associated pipelines and challenges. But even if we are not going to detail, not going to know about the details of different platforms, we'll focus on transcriptomics also. But these are overlapping. These challenges are overlapped across all these techniques. Okay, so and then we will focus on the limitations of the transcriptomics and requirement of multi-omics integration. So next question is, we'll see the challenges, but before that the question is why we need multi-omics integration. What are the limitations of transcriptomics? What do you think? Just now I said this is a very good technique and we can use this technique to identify different genes. So is it the one which we actually require? Or is there any problem with that thing? What do you think? What are the limitations?
Sir, a large data is found which is difficult to read.
Large data is found which is difficult to read. What do you
Yes,
mean by
sir.
that? What do you mean by that? Can you explain why it is difficult to read?
Sir, as we get the large data, so it is the complexity is enhancing. So
Okay. So you are saying that it is hard to analyze that large data. But then if that is the limitation, so if you use multi-omics, whether it will reduce, because multi-omics is more complex. Okay, multi-omics will be more complex, isn't it? So multi-omics means you can integrate transcriptomics with proteomics, transcriptomics with genomics, and then you can get the specific hits or interested molecules.
so it is cost. It is not cost effective.
Which one? Transcriptomics? No,
Yes.
think logically. Transcriptomics is a single technique. So whatever cost it will take, obviously, if you are planning to perform multi-omics technique, so it will cost more. It will cost more. Even though sometimes we required to integrate different kinds of omics technologies. The question is why? Okay, now I am giving you one easy example, like if you are focusing on transcriptomics. Okay, now you are getting the list of RNAs which are expressing in a particular sample. Fine. But if the disease is causing due to the presence or absence, or what I say, due to the higher expression of that particular gene, that means in terms of protein. Okay, so this transcriptomics only, you cannot identify that particular protein, right? Because it is only showing the list of RNA, not the protein. And if the disease is caused by some proteins, so then you cannot correlate that the RNA expression is high, that means the protein expression will also be high, isn't it? What do you think? If I am saying that in an experimental study you have found 200 of genes are overexpressed than the control sample, now can you say that that 200 proteins will be also upregulated in that sample when you will study the protein? What do you think? Anyone? Yes or no? What do you think? If the RNA expression is high, so protein expression will be also high? Yes or no?
Yes.
Okay, I got one answer, yes. So everybody is agreeing with him.
So not always true. It's
Not always, yes, sir. Because if some condition is applied, then it will be decrease, or maybe it
I
depends the
think
condition, sir.
Okay, so you are saying that condition. Any
I,
other? Yes.
I think it will be more higher because from the previous explanation you made, you said if the analysis is the data, the input data is small, the output data will be more higher. Is it?
No, 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 no. That is for explanation of high throughput techniques. But when I am talking about the expression of genes and expression of proteins, so those are different. Okay, so for quantifying this expression of RNA or expression of protein, you can use high throughput techniques. And to explain that technique, I told that if you are using some small sample, you will get large amount of data from that sample. That is totally different. But here, what I am trying to take that even if you are getting that some RNA expression, some RNAs are expressed in a sample in more amount rather than control sample, then also you cannot say that those proteins also will be in higher amount in that sample, because the RNA expression and the protein expression is not directly correlated. Okay, there are many different steps by which the expression levels of proteins can be changed. Okay, after post translational event, or even post transcriptional event, or post translational event, some changes, some modifications will be there, and that's why that particular protein what you are expecting for the upregulation of the RNA transcript, so that may not happen. Okay, so that is the problem. You cannot correlate the expression level of RNA and expression level of protein. So that is one major difficulties in transcriptomic analysis. Okay. Okay, so today's class time is over. So we will meet again tomorrow.